Welcome to Molecular Plant Science. Please like, subscribe, and share. Welcome to our exploration of protein subcellular prediction, a fascinating area of bioinformatics that plays a crucial role in understanding gene function in plants. Today, we'll focus on a powerful tool called WolfSort. This software is designed to predict the subcellular localization of proteins, which is essential for determining their potential functions within the cell. Why is this important? Well, proteins don't just float around aimlessly. They have specific roles based on where they are located. For instance, proteins found in chloroplasts are likely involved in photosynthesis, while those in the nucleus might play a role in gene regulation. By using WolfSort, researchers can predict where a protein is likely to reside, providing valuable insights into its function. Now, let's connect this to gene family analysis. Gene families are groups of related genes that often share similar functions. In plants, understanding these families can illuminate how they adapt to various environments, resist diseases, or respond to stress. By applying WolfSort in a genome-wide analysis, scientists can identify all the members of a gene family and predict their subcellular locations. This comprehensive approach enables researchers to hypothesize about the roles of these proteins based on their predicted localizations. For example, if a gene family member is predicted to be localized in the mitochondria, we can infer it may be involved in energy production. Conversely, if it's found in the cell membrane, it might play a role in signaling or transport. This predictive power not only enhances our understanding of plant biology, but also aids in the development of crops with improved traits. In summary, protein subcellular prediction using WolfSort is a vital step in unraveling the complex functions of gene family members in plants, paving the way for advancements in agriculture and sustainability.
Subcellular localization plays a critical role in genome-wide analyses by providing insights into the functional roles of genes within specific cellular contexts. By determining where proteins encoded by gene families are localized, such as the nucleus, cytoplasm, mitochondria, or chloroplasts, it becomes possible to predict their functional roles based on their cellular environment. This approach is particularly valuable in studying large gene families, as it enables researchers to group genes based on their subcellular destinations, aiding in functional annotation and the identification of related pathways. For example, genes encoding proteins targeted to the chloroplast may be grouped and studied for roles in photosynthesis or stress response, whereas nuclear-localized proteins might be linked to transcriptional regulation. Through the integration of experimental data, such as fluorescent tagging and computational tools, subcellular localization facilitates a comprehensive understanding of gene function across entire families, accelerating discoveries in fields such as functional genomics, systems biology, and crop improvement.
Subcellular localization analysis is a pivotal aspect of genome-wide studies, offering critical insights into gene function by linking protein destination within the cell to its potential biological roles. The cellular compartmentalization of proteins, determined through localization studies, helps unravel complex cellular processes and biological pathways. In large gene families, such as transcription factors or enzymes, subcellular localization allows the classification of family members based on their specific cellular niches. For instance, proteins localized to the chloroplast may be involved in photosynthesis or reactive oxygen species management, while those targeted to the mitochondria could be associated with energy metabolism or apoptosis. By leveraging advanced techniques such as fluorescence microscopy, bioinformatics tools, and machine learning-based predictors, researchers can systematically annotate subcellular localization across entire genomes. This approach aids in grouping gene family members with similar localizations, thereby hypothesizing shared or complementary functions. Moreover, subcellular localization provides a framework for investigating protein-protein interactions and regulatory networks, as proteins are more likely to interact within the same compartment. In functional genomics, subcellular localization bridges the gap between gene sequence and biological activity, offering an additional layer of validation to computational predictions. For crop improvement and pathogen resistance research, This knowledge is invaluable in identifying candidate genes for breeding programs, especially when targeting traits related to stress tolerance, metabolism, or defense mechanisms. Thus, the integration of subcellular localization data into genome-wide analysis enhances our understanding of gene functions and their coordination in maintaining cellular and organismal homeostasis. Subcellular localization is a cornerstone of genome-wide analysis, providing a critical lens through which researchers can interpret gene functions within the spatial and functional landscape of the cell. Proteins are not randomly distributed. Their specific cellular destinations, such as the nucleus, mitochondria, endoplasmic reticulum, Golgi apparatus, or chloroplasts, are tightly linked to their roles. By studying where proteins encoded by genes are localized, researchers can infer their functional roles, predict biological pathways, and explore regulatory networks. This spatial information is particularly vital in analyzing large gene families, where diverse functions may be hidden under shared sequence motifs. Localization patterns help segregate these families into functional groups, unraveling the complexity of their roles within cellular systems. Advanced technologies, including high-resolution fluorescence microscopy, proteomics-based organelle fractionation, and computational predictors like DeepLoc and WolfSort, allow high-throughput and precise determination of protein localization. These methods enable researchers to analyze gene function at a systems level, correlating localization data with transcriptomic, proteomic, and interactomic datasets. For instance, nuclear localized proteins might reveal roles in transcription regulation, while those in the plasma membrane often relate to signal transduction or transport processes. Similarly, plastid localized proteins are typically linked to photosynthesis, lipid biosynthesis, or stress responses, highlighting potential targets for improving crop performance. Grouping gene family members based on subcellular localization offers several benefits. It refines functional predictions by excluding proteins from irrelevant pathways, identifies redundancy within gene families, and helps highlight key players within specific compartments. This is particularly useful in model organisms and crops, where understanding subcellular roles can guide genetic engineering strategies. In plant breeding, subcellular localization is critical for identifying genes involved in resistance mechanisms. For instance, proteins localized to the apoplast may play roles in pathogen defense, while those in peroxisomes might manage oxidative stress. Furthermore, subcellular localization helps decode evolutionary adaptations. The expansion of certain gene families in specific compartments, such as the chloroplast or vacuole, often reflects an organism's adaptation to environmental challenges. For example, in plants, localization of secondary metabolite enzymes in vacuoles or defense proteins in the cell wall reflects their roles in survival and fitness. By integrating subcellular localization data into genome-wide studies, researchers can develop more holistic models of cellular organization and functionality, paving the way for advancements in functional genomics, systems biology, and applied agricultural sciences.
A heat map is a data visualization tool that uses a matrix-like structure with color gradients to represent data values. The intensity or shade of the color corresponds to the magnitude of the data points, making it easier to identify patterns, trends, and relationships within large datasets. In biological studies, heat maps are widely used to represent data, such as gene expression levels, genetic variations, epigenetic modifications, or protein-protein interactions.
Applications of heat maps in genome-wide analysis. Gene expression profiling. Heat maps are extensively used to visualize the expression levels of thousands of genes across different conditions, tissues, or time points. For example, in RNA-seq data analysis, heat maps can highlight differentially expressed genes, DGs,